Diversified DNA libraries for antibody, peptide, and nanobody library construction, methods and techniques. Narrated by Luke McLaughlin, biotech digital marketer, business developer, and life science content creator. Generating libraries of diversified oligonucleotides, especially those with sequences containing randomized codon regions, is a critical step in the development of antibody, peptide, and nanobody libraries. These libraries are foundational tools in therapeutic discovery, enabling the identification of molecules with high specificity and affinity for a wide range of targets. The methods or technologies used for generating these libraries have evolved to allow high levels of diversity, specificity, and efficiency. The currently available methods and techniques for generating such libraries are solid-phase DNA synthesis, trimer phosphoramidite-based synthesis and considerations regarding trimer phosphoramidite technology, PCR-based random mutagenesis, DNA shuffling, microchip-based DNA synthesis, oligonucleotide ligation, CRISPR-Cas9 mediated library generation, cell-free transcription translation systems, phage display with randomized peptide libraries. All these will be discussed in detail in this podcast, after which library cloning, characterization, and functionalization will be considered. Solid-phase DNA synthesis, phosphoramidite technology for antibody library generation. This traditional method involves the stepwise addition of nucleotides to a growing chain immobilized on a solid support. It can be adapted to introduce randomization at specific positions by mixing different phosphoramidites in controlled ratios during the synthesis cycle, allowing for the generation of libraries with diversified sequences. Advantages: precision in randomization. SPDS allows for precise control over the location and degree of randomization within the codon regions. This precision is critical for creating libraries with a specific scope of diversity, ensuring that all possible variations of interest are represented. High throughput. The method is amenable to automation, allowing for the simultaneous synthesis of multiple oligonucleotides. This high-throughput capability is essential for generating large libraries with extensive diversity in an efficient manner. Customization. It offers the flexibility to incorporate various modifications, such as fluorescent labels or different types of linkers, which can be valuable for downstream applications like sorting or identification of the library components. Scalability. Libraries can be synthesized on a scale that matches the requirements of the application, from small-scale exploratory studies to large-scale screens. Disadvantages: complexity and cost. The synthesis of complex libraries with high degrees of randomization and large size can become prohibitively expensive. Costs escalate with the complexity of the library due to the need for specialized reagents and the increased synthesis time. Error rate. The fidelity of SPDS decreases as the length of the oligonucleotide increases, leading to errors in the library. For randomized regions, this issue is compounded as the synthesis involves mixing bases at specific positions, which can lead to incorrect incorporations and affect the diversity and utility of the library. Limited length. SPDS is typically efficient for oligonucleotides up to a certain length, usually around 200 nucleotides. For libraries requiring longer sequences with randomized regions, this limitation can be a significant barrier, potentially necessitating the use of additional techniques to assemble longer sequences from shorter oligos. Purification challenges. Post-synthesis, the library requires purification to remove incomplete or erroneous sequences. The purification process can be challenging and inefficient for complex libraries, potentially resulting in loss of diversity or yield. Environmental impact. The synthesis process uses large quantities of chemicals and solvents, some of which are hazardous. The disposal and environmental impact of these materials require careful management and can add to the overall cost and complexity of library generation. Trimer phosphoramidite-based synthesis. As detailed previously, trimer phosphoramidite-based synthesis allows the addition of 3 nucleotides at a time, with specific trimer blocks designed to introduce desired randomizations. This method offers a balance of speed, efficiency, and the ability to generate complex libraries with precise control over the degree of sequence diversity. Advantages: high specificity and efficiency. Trimer phosphorimidites enable the synthesis of codons with specific nucleotide sequences, allowing for precise control over the amino acid sequence variability in the resulting peptide or protein library. This specificity is crucial for applications requiring a high degree of control over sequence composition. Reduced complexity. By synthesizing trimer blocks that correspond to specific amino acids, the complexity of the library is reduced. This is because each trimer can encode for a specific amino acid, reducing the occurrence of stop codons and non-functional sequences, compared to libraries synthesized using individual nucleotides. Faster synthesis of large libraries. The use of trimer phosphorimidites can expedite the synthesis process because each addition corresponds to 3 nucleotides instead of 1, allowing for quicker assembly of the desired sequences, cost-effective for certain applications. When the goal is to incorporate specific variations or cover a defined set of sequences, trimer phosphorimidite-based synthesis can be more cost-effective than methods that introduce randomization at the individual nucleotide level. Disadvantages. Limited diversity. While trimer phosphorimidites provide specificity, they can also limit the diversity of the library. Since each trimer corresponds to a specific codon, the range of possible mutations at each position is constrained by the trimers used in the synthesis. Complexity in synthesis and design. Designing and synthesizing specific trimer phosphorimidites can be complex and time-consuming. The process requires careful planning to ensure that the desired diversity is achieved without introducing unwanted biases. Higher initial costs. The initial cost of synthesizing specific trimer phosphorimidites can be higher than using standard nucleotide phosphorimidites, especially for custom libraries where unique trimers are required. Risk of synthesis errors. Although trimer phosphorimidite-based synthesis can be highly efficient, the risk of synthesis errors increases with the length of the oligonucleotide. Errors in trimer incorporation can lead to sequence heterogeneity and reduce the overall quality of the library. Limited availability. Not all trimer phosphorimidites may be commercially available, limiting the ability to synthesize certain sequences or requiring the custom synthesis of specific trimers, which can be costly and time-consuming. Considerations regarding trimer phosphorimidite technology. The essence of trimer phosphorimidite technology. Trimer phosphorimidite technology represents a sophisticated approach in synthetic biology for the assembly of DNA sequences. Phosphorimidites are chemical compounds used in the synthesis of oligonucleotides, short sequences of nucleic acids, which are the building blocks of DNA. Traditional phosphorimidite chemistry involves adding nucleotides one at a time in a predefined sequence to synthesize a specific DNA molecule. However, trimer phosphorimidite technology revolutionizes this process by allowing the simultaneous addition of 3 nucleotides corresponding to an amino acid codon, thus significantly accelerating the synthesis of oligonucleotides and enhancing the efficiency of generating diverse sequences. Integration with oligonucleotide libraries and randomized codons. The integration of trimer phosphorimidite technology with oligonucleotide libraries containing randomized amino acid codons is a critical leap forward. By employing trimers that represent all possible codons for the 20 natural amino acids, scientists can create libraries of oligonucleotides that, when translated, cover an extensive range of amino acid sequences. Randomization of codons in these libraries is crucial as it allows for the generation of a vast diversity of peptides, which can then be used to create corresponding antibody fragments. The process involves designing oligonucleotide sequences that include regions of randomization, where each position can incorporate any of the trimer phosphorimidites. This approach enables the synthesis of DNA sequences that encode a wide array of potential antibody variable regions, which are critical for binding to antigens. The randomized sequences are then inserted into vectors and expressed in a suitable host, such as bacteria or yeast, to produce a library of antibody fragments. The sequence design of these oligonucleotides must be carefully considered to ensure successful cloning, expression, and functionality of the product. One of the key considerations is avoiding restriction sites that could interfere with cloning processes or vector stability. Below, we discuss these considerations in more detail, particularly focusing on avoiding restriction sites and listing common enzymes and their restriction sites to avoid in the codon design. Sequence design considerations for optimal expression. Codon optimization. This involves modifying the DNA sequence to use codons preferred by the host organism, which can enhance translation efficiency and protein expression levels. Avoidance of internal promoters. Sequences that could act as cryptic promoters within the coding sequence may lead to premature transcription termination or unwanted protein expression patterns. GC content. An appropriate GC content is crucial for stability and efficient transcription and translation. Extremely high or low GC content can lead to issues with DNA synthesis, cloning, and expression. Secondary structure prevention. Sequences prone to forming strong secondary structures, such as hairpins, can hinder transcription and translation processes. Tools for predicting RNA secondary structure can be used to identify and modify such sequences. Avoidance of repetitive sequences. Repetitive sequences can lead to recombination events, instability in the vector, and challenges in sequencing the inserts. Avoiding restriction sites. One of the paramount considerations is to design oligonucleotide sequences that avoid restriction enzyme recognition sites used in the cloning process or present in the multiple cloning site, MCS, of the vector. This prevents unwanted cleavage of the insert or vector, which can complicate cloning and downstream applications. When designing oligonucleotides or modifying sequences, bioinformatics tools can help identify and eliminate these restriction sites. Importance of avoiding restriction enzyme sites. Preservation of library diversity. Restriction enzyme sites within the oligonucleotide sequences can lead to cleavage of the DNA, resulting in the loss of potential antibody variants. This reduction in diversity can significantly impair the library's effectiveness in screening for antibodies with the desired properties. Unintended cloning complications. The presence of restriction enzyme sites can complicate the cloning process. These sites may interfere with the ligation of oligonucleotides into vectors, potentially leading to incorrect insertions or deletions that can disrupt the expression of functional antibodies. Efficiency and cost. Avoiding restriction sites can increase the efficiency of library construction and screening, reducing both the time and cost associated with identifying promising antibody candidates. Sequence integrity. Ensuring that restriction sites are absent or strategically placed helps maintain the integrity of the sequences within the library, allowing for accurate representation and expression of the intended antibody variants. Common restriction enzymes and sites to avoid. Here's a list of some common restriction enzymes used in molecular biology and the recognition sequences that should be avoided in the codon design for antibody libraries. EcoRI, recognition site, GATTC. BAM-HI, recognition site, GATCC. HIND-II, recognition site, AAGCTT. NOTI, recognition site, GCGGECGC. XHOI, recognition site, CTCGIG. SACI, recognition site, GGCTC. KPNI, recognition site, GGTAC. SMAI, recognition site, CCCCGGG. XBI, recognition site, TCTAGA. SPI, recognition site, ACTAGT. When designing synthetic oligonucleotides for insertion into an expression vector, it's crucial to screen the sequence not only for the presence of these common restriction sites, but also for less frequently used enzymes that might be relevant to the specific cloning strategy or vector in use. Strategies to avoid restriction sites. Silent mutations. Introducing silent mutations to alter the DNA sequence without changing the encoded amino acid can eliminate unwanted restriction sites. Alternative cloning strategies. Techniques such as Gibson assembly or topo registered trademark cloning can be employed, which do not rely on restriction enzymes for cloning, thereby circumventing the issue. Custom enzyme selection. When possible, choose restriction enzymes that do not have recognition sites within the gene of interest, or design the construct such that critical regions do not coincide with these sites. Generating antibody libraries with large epitopal diversity. The primary advantage of using trimer phosphoramidite technology in conjunction with randomized oligonucleotide libraries is the ability to generate antibody libraries with unprecedented epitopal diversity. Epitopes are the specific parts of an antigen molecule to which an antibody attaches itself, and diversity in epitopes is essential for creating antibodies capable of recognizing and binding to a broad spectrum of antigens. The large-scale diversity achieved through this method ensures that the resulting antibody library contains candidates with high affinity and specificity for a wide range of targets. This is particularly beneficial for therapeutic applications where the identification of antibodies that can precisely target disease-associated antigens is crucial. Moreover, the diversity of the library increases the likelihood of discovering antibodies with novel mechanisms of action, or those that can bind to challenging targets, such as conserved regions of viruses or cancer cells. PCR-based random mutagenesis, PCR, polymerase chain reaction, can be used to introduce random mutations across a given sequence or at specific sites. Techniques such as error-prone PCR, degenerate oligonucleotide PCR, and DNA shuffling allow for the creation of highly diversified libraries by varying the fidelity of the DNA polymerase or the composition of the oligonucleotide primers. Advantages. High diversity. PCR-based random mutagenesis can generate a high degree of nucleotide sequence diversity. This allows for the exploration of a vast sequence space within the oligonucleotide library, which is beneficial for identifying novel sequences with desired properties. Flexibility and simplicity. The method is flexible, allowing for the introduction of mutations across a wide range of frequencies depending on the experimental conditions, for example, type of polymerase, MgCl2 concentration, nucleotide analogs. It's also relatively simple to perform, requiring standard PCR equipment and reagents. Cost effectiveness. Compared to some other methods for generating diversity, PCR-based random mutagenesis can be more cost-effective, especially for initial exploratory studies where a wide range of sequences is desired without the need for specific sequence modifications. Rapid library generation. PCR can rapidly amplify target sequences, enabling quick generation of large libraries. This rapid turnaround is essential in iterative processes like directed evolution. Disadvantages. Bias in mutation. Introduction. PCR-based mutagenesis can introduce biases in the types and locations of mutations due to the intrinsic properties of the polymerases used and the specific conditions of the reaction. This can limit the uniformity of mutation distribution across the library. Error propagation. Errors introduced during early cycles of PCR can be propagated throughout the library, leading to over-representation of certain mutants. This can skew the diversity of the library and affect the outcome of selection experiments. Limited control over mutation rate. While conditions can be adjusted to influence the overall mutation rate, controlling the exact rate of mutation and ensuring that it is uniformly distributed across all positions in the sequence can be challenging. Potential for chimeric sequences. PCR conditions that favor recombination can result in chimeric sequences, especially in the presence of similar sequences. This can complicate the interpretation of results from libraries intended for the study of specific sequence variants. Requirement for subsequent cloning and transformation. After PCR-based mutagenesis, the mutated sequences often need to be cloned into vectors and transformed into host cells for expression and screening. This additional step can introduce complexity and variability into the library. DNA shuffling. DNA shuffling involves the fragmentation of a pool of DNA sequences, followed by their random reassembly through PCR. This method can create significant diversity within libraries by recombining different sequences, including those with randomized regions. Advantages. Rapid generation of diversity. DNA shuffling can create a high degree of genetic diversity by recombining mutations from different variants. This allows for the exploration of a vast sequence space more efficiently than point mutagenesis. Integration of beneficial mutations. By recombining sequences, beneficial mutations from different variants can be combined into a single molecule, potentially leading to synergistic improvements in the desired traits. Accelerated evolutionary process. DNA shuffling mimics the natural evolutionary process of recombination, but at an accelerated pace, enabling rapid improvement of molecular functions. Reduced risk of detrimental mutations. Compared to methods that introduce random mutations across the entire sequence, DNA shuffling can reduce the accumulation of detrimental mutations, since it often uses parent sequences that are already functional. Flexibility. It can be applied to a wide range of DNA sequences, from small oligonucleotides to large genomic regions, and is not limited to sequences with known functions. Disadvantages. Requirement for initial sequence diversity. DNA shuffling requires a set of related sequences as a starting point, which can limit its use to cases where such diversity is available or can be generated. Complexity of the recombination process. The process involves several steps, including DNA fragmentation, random reassembly, and sometimes cloning into vectors for expression. These steps add complexity and can introduce variability in the outcome. Potential for biased recombination. The recombination process might favor certain sequences over others, potentially leading to a biased representation of variants in the library. Difficulty in controlling shuffle. Intensity, controlling the degree of shuffling and ensuring uniform recombination across the entire library can be challenging. This might result in uneven distribution of diversity. Potential loss of function. While the method aims to combine beneficial mutations, the random nature of recombination can sometimes result in non-functional sequences, especially if key structural or functional domains are disrupted. Screening and selection challenges. The high diversity and potential complexity of the resulting libraries require efficient screening and selection strategies to identify variants with the desired properties. Microchip-based DNA synthesis. Microchip-based technologies allow for the parallel synthesis of a vast number of oligonucleotides on a single chip, with each spot synthesizing a unique sequence. This method can generate highly diverse libraries by designing chips to synthesize oligonucleotides with randomized regions. Companies like Twist Bioscience and Agilent Technologies offer services based on this approach. Advantages. High throughput. Microchip-based synthesis allows for the parallel generation of a vast number of oligonucleotides on a single chip, significantly increasing throughput compared to traditional column-based synthesis. Cost efficiency. By synthesizing many sequences in parallel, the cost per sequence can be dramatically lower, making large-scale studies and the generation of extensive libraries more financially feasible. Scalability. The technology is highly scalable, allowing for the production of increasingly large libraries as the technology advances and fabrication techniques improve. Precision and flexibility in library design. Microchip-based synthesis can precisely control the sequence of each oligonucleotide, allowing for the design of complex libraries with specific patterns of randomness or variation in codon regions. Rapid prototyping. The method supports rapid iteration and prototyping of genetic constructs, enabling quick testing of hypotheses in synthetic biology and genetic engineering. Disadvantages. Synthesis error rates. As with any chemical synthesis process, Errors such as deletions, insertions, and substitutions can occur, potentially introducing unwanted variability into the library. Sequence length limitations. Current microchip-based synthesis technologies may have limitations on the length of DNA that can be synthesized with high fidelity. Longer sequences might require additional steps, such as assembly from shorter oligonucleotides, which can introduce further complexity and potential for errors. Complexity of library construction. While the synthesis step is efficient, constructing functional libraries from synthesized oligonucleotides often requires additional steps, including cloning into vectors and transformation into host cells, each adding potential points of failure or inefficiency. Access and cost for small projects. While the cost per sequence is low for large-scale projects, the initial setup cost and access to the technology may still be prohibitive for smaller laboratories or projects with limited budgets. Handling and processing of large datasets. The generation of vast libraries entails dealing with large datasets, requiring computational tools for design, error correction, and analysis, which might be a challenge for researchers without access to bioinformatics support. Oligonucleotide ligation. This method involves the ligation, joining, of short synthesized oligonucleotides into longer sequences. By using oligonucleotides that contain randomized regions, it's possible to assemble diversified libraries. The ligation can be facilitated by enzymes like DNA ligase or by using chemical ligation methods. Advantages. Precision in library construction. Ligation allows for the precise assembly of sequences with specific randomized regions. By designing oligonucleotides that contain randomized codons flanked by defined sequences, researchers can create highly targeted libraries. Flexibility in design. This method offers flexibility in incorporating various modifications, such as phosphorylation or fluorophores, into the oligonucleotides before ligation, enabling the creation of functionally diverse libraries. Compatibility with various templates. Oligonucleotide ligation can be used to assemble libraries on both DNA and RNA templates, making it suitable for a wide range of applications, including studies on RNA functionality and RNA-protein interactions. High fidelity. Ligation can achieve high fidelity in the assembly process, particularly when using high-quality oligonucleotides and optimized conditions, leading to accurate representation of the intended library diversity. Disadvantages: complexity and time-consuming. The process can be complex and time-consuming, especially for large libraries, since each ligation step needs to be controlled and verified. The efficiency of ligation decreases as the number of fragments to be ligated increases. Dependency on efficient ligation. The success of library construction heavily relies on the efficiency of the ligation reaction, which can vary based on the oligonucleotide sequences, lengths, and the ligase enzyme used. Potential for ligation bias. There can be a bias in ligation efficiency towards certain sequences or structural motifs, which might result in uneven representation of sequences in the final library. Sequence length limitations. While effective for shorter sequences, the efficiency of assembling very long sequences through ligation can be limited. This might necessitate additional steps, such as PCR amplification or cloning, to generate the final library constructs. Cost. Depending on the complexity of the library and the number of oligonucleotides required, the cost of synthesizing high-quality oligonucleotides and the enzymes needed for ligation can be significant, especially for high-throughput or large-scale projects. Need for post-ligation processing. After ligation, additional steps are often required to transform the ligated products into a usable library, including cloning into vectors and transformation into host cells, which adds further complexity and potential points of failure. CRISPR-Cas 9-mediated library generation. Although primarily known for gene editing, CRISPR-Cas9 technology can be adapted to generate diversified libraries. Guide RNAs, gRNAs, can be designed to target specific genomic locations, with the introduction of diversified sequences achieved through the repair of Cas9-induced double-strand breaks using oligonucleotides containing randomized regions. Advantages: high precision and specificity. CRISPR-Cas9 allows for targeted modifications with high precision at specific genomic locations. This enables the creation of libraries where variations are introduced exactly where desired, minimizing off-target effects. Versatility in targeting. The system can be designed to target virtually any genomic location, provided there is a suitable protospacer-adjacent motif, bosenane, site, allowing for the generation of diverse libraries across different genes or regulatory elements. Efficient library construction. By delivering a pool of guide RNAs, gRNAs, along with a template for repair, a wide array of mutations, including insertions, deletions, and substitutions, can be introduced simultaneously, facilitating efficient library construction. Direct in vivo application, CRISPR-Cas. 9 libraries can be used directly in cells, including human cells, for functional genomic studies, drug screening, and more, bypassing the need for in vitro cloning steps. Facilitates high-throughput screening. Combined with next-generation sequencing, NGS, and other high-throughput analysis techniques, CRISPR-Cas9 libraries enable the rapid screening of genetic variants for functional studies. Disadvantages: off-target effects. Despite high specificity, CRISPR-Cas9 can still induce off-target mutations, potentially confounding the interpretation of library screening results. PAM site. Requirement: the necessity for a PAM sequence near the target site can limit the possible target locations for editing, although newer CA's variants with different PAM requirements are expanding these capabilities. Efficiency, variability. The efficiency of CRISPR-Cas9 editing can vary significantly depending on the target site, the cell type, and the delivery method of the components, affecting the uniformity of the library. Technical complexity. The design, optimization, and delivery of CRISPR-Cas9 components, gRNAs, Cas9, and repair templates require specialized knowledge and resources, which can be a barrier for some laboratories. Regulatory and ethical considerations. Especially relevant for in vivo applications, there are ethical and regulatory considerations that must be navigated, particularly for any potential therapeutic applications arising from library screening. Need for effective delivery systems, efficient delivery of CRISPR-Cas9 components into cells, especially for in vivo applications, remains a challenge and is critical for successful library generation and screening. Cell-free transcription translation systems. These systems can be used to directly express oligonucleotides containing randomized codon regions into functional peptides or proteins. By linking the in vitro synthesis of DNA to its immediate expression, this method allows for the rapid screening and selection of functional molecules from a diversified library. Advantages: rapid protein synthesis. Cell-free systems can synthesize proteins within hours, significantly faster than cell-based expression systems, facilitating quick screening of oligonucleotide libraries. Simplified setup. Without the need for cell culture, the setup is simpler and can be more amenable to high-throughput formats, enabling the screening of large libraries without the complexities of cell maintenance. Direct control over reaction conditions. The researcher can directly control the environment, such as the concentration of ions, energy sources, and cofactors, which can be advantageous for expressing proteins that require specific conditions or for incorporating non-standard amino acids. Avoidance of cellular toxicity. Since the system is cell-free, toxic proteins or those that might interfere with cellular viability can be expressed and studied without harming living cells. Functional screening without cloning. Libraries of DNA templates can be directly used for protein synthesis without the need for cloning into expression vectors and transformation, streamlining the library screening process. Disadvantages: protein folding and solubility issues. Cell-free systems may not always support the correct folding of complex proteins or the formation of multi-subunit complexes, potentially limiting the functional analysis of some proteins. Limited protein yield. While sufficient for many analytical purposes, the protein yields from cell-free systems are often lower than what can be achieved with in vivo systems, which might be a limitation for applications requiring large amounts of protein. Cost, depending on the scale and the source of the cell-free extract, for example, commercially prepared versus lab-made, the cost can be higher than for traditional in vivo expression systems, especially for high-throughput screening of large libraries. Durability of the reaction. Cell-free systems can lose activity over time, limiting the duration of protein synthesis. Continuous or long-term expression is more challenging compared to living cells. Lack of post-translational modifications. Many cell-free systems, especially those derived from bacteria, cannot fully replicate the post-translational modifications found in eukaryotic cells, which can be crucial for the activity of some proteins. Phage display with randomized peptide libraries. Phage display technology can be adapted to express peptides or proteins from randomized oligonucleotide libraries on the surface of bacteriophages. This method combines the generation of diversity at the DNA level with the direct selection of molecules based on their binding affinity to a target. Advantages: high throughput screening. Phage display allows for the screening of millions to billions of peptides or proteins simultaneously against a target, enabling the identification of high-affinity binders from a vast sequence space. Direct linkage between phenotype and genotype. The peptide or protein of interest displayed on the phage surface is directly linked to the DNA inside the phage that encodes it. This linkage facilitates easy identification and replication of successful binders. Versatile target range. The targets can be virtually any molecule, including proteins, peptides, DNA, RNA, small molecules, and even whole cells, making phage display a versatile tool for various applications in research, therapeutics, and diagnostics. In vitro selection. Since selections are performed in vitro, phage display is not limited by the physiological constraints of cellular expression systems, allowing the discovery of binding partners for toxic or otherwise challenging targets. Cost-effectiveness. Phage display does not require expensive equipment or reagents, making it a cost-effective option for generating and screening peptide libraries. Disadvantages. Bias toward phage-compatible sequences. Not all peptides or proteins are suitable for display on the phage surface. Some may affect phage viability or display efficiency, leading to a selection bias. Limited by phage biology. The size of the displayed peptide or protein can be limited by the phage biology, as large inserts can disrupt phage assembly or function. Complexity of library construction. Constructing a high-quality library with a diverse range of randomized codons can be technically challenging and time-consuming. Potential for non-specific binding. Non-specific binders can be enriched during the selection process, requiring careful design of control and washing steps to minimize their impact. Scale-up challenges. While phage display is excellent for identifying high-affinity binders, translating these findings into practical applications, for example therapeutic development, requires further steps, including validation in vivo and scale-up production, which can be challenging and resource-intensive. Need for multiple rounds of selection. To isolate high-affinity binders, several rounds of selection, biopanning, are often necessary, which can be time-consuming and may not always result in the identification of desired targets. Library cloning, characterization, and functionalization. Selection of expression vector. Vector choice. An appropriate expression vector is chosen based on the host system, for example bacterial, yeast, mammalian, and the desired application, for example protein production, functional studies. Considerations include the vector's origin of replication, antibiotic resistance marker, promoter, and multiple cloning site, MCS. Preparation. The vector is prepared for cloning by digestion with restriction enzymes that cut at sites flanking the MCS, or using a method that does not rely on restriction sites, such as Gibson assembly or TOPO registered trademark cloning. Preparation of oligonucleotides for cloning. Annealing. If the oligonucleotides are single-stranded, complementary strands are annealed to form double-stranded DNA. This is typically done by mixing equimolar amounts of each strand, heating the mixture, and then allowing it to cool slowly. End modification. The double-stranded oligonucleotides are modified if necessary to facilitate cloning. This can include the addition of phosphate groups to the 5 ends, phosphorylation, or the addition of overhangs compatible with the prepared vector. Ligation into the vector, mixing. The prepared oligonucleotides are mixed with the prepared vector in the presence of a DNA ligase enzyme. The ligase catalyzes the formation of phosphodiester bonds between the ends of the oligonucleotides and the vector, integrating the insert into the vector. Conditions. Ligation conditions, temperature, time, and buffer composition are optimized for the highest efficiency. Transformation into host cells. Preparation of competent cells. Host cells, often E. coli for bacterial expression systems, are made competent to take up DNA through chemical treatment or electroporation. Introduction of recombinant vector. The ligation mixture is introduced into the competent cells, which are then incubated under conditions that allow for the uptake of the DNA. Selection and screening. Selection. Transformed cells are plated on agar medium containing an antibiotic that selects for cells containing the antibiotic resistance gene from the vector. Only cells that have successfully taken up and maintained the vector will grow. Screening. Colonies are screened for the presence of the insert using colony PCR, restriction digestion, or sequencing. This confirms that the oligonucleotide has been correctly integrated into the vector. Expression testing. Induction of protein expression. Conditions for inducing protein expression from the promoter in the vector are optimized and applied to the selected clones. Verification. The expression of the recombinant protein is verified using techniques such as SDS page and Western blotting. Scale-up and protein purification. Culturing. Clones confirmed to express the protein of interest are cultured under optimized conditions to produce larger quantities of the protein. Purification. The recombinant protein is purified from the culture using methods such as affinity chromatography, taking advantage of tags that may have been included in the design of the oligonucleotide. Initial screening. Target antigen preparation. The target antigen against which the antibodies are to be screened is prepared and, if necessary, labeled for detection. The preparation method depends on the screening technique to be used. Initial screening, panning, phage or yeast display. The library is exposed to immobilized antigen. Non-binding fragments are washed away, and bound fragments are eluted and amplified for further rounds of selection. Fluorescence-activated cell sorting, FXS, used primarily with yeast or bacterial display, where cells displaying antibody fragments that bind to the antigen are sorted based on fluorescent signals. Cell-based screening. If the target is a cell surface antigen, screening can be directly performed on cells expressing the antigen. Enrichment and amplification. After initial screening, selected clones are amplified. For phage display, this involves infecting bacteria with the selected phages to produce more of the selected antibody fragments for subsequent rounds of selection. For yeast or bacterial display, selected cells are cultured under conditions that promote growth and expression of the antibody fragments. Secondary screening. High-throughput binding. Assays. Techniques such as ELISA are used to quantitatively assess the binding affinity of individual antibody fragments to the antigen. Functional assays. Depending on the intended application, selected antibodies may be screened for functional properties, such as neutralization activity or receptor blocking. Sequence analysis and cloning. Antibody fragments that pass secondary screening are sequenced to identify the genetic makeup of the variable regions. Selected sequences are then cloned into expression vectors for further characterization and production. Affinity maturation optional. If higher affinity is required, selected antibody fragments may undergo affinity maturation processes, such as directed evolution or site-directed mutagenesis, followed by additional rounds of screening. Expression and purification of selected antibodies. Clones with the desired properties are expressed in a suitable system, for example, E. coli, yeast, mammalian cells, for large-scale production. Recombinant antibodies are purified using techniques appropriate for the expression system and antibody format, for example, affinity chromatography. Final characterization. Purified antibodies are subjected to thorough characterization, including determination of binding affinity, specificity, and in vitro and in vivo functional assays to confirm their therapeutic potential. Sequence analysis. Objective: to determine the genetic sequences of the variable regions of selected antibody fragments. Process. Antibody fragments that demonstrated desirable binding and screening processes are sequenced using methods like Sanger sequencing or next-generation sequencing, NGS, to identify their variable region sequences. Affinity and specificity assessment. Objective: to evaluate the binding strength and specificity of the antibody fragments towards the target antigen. Process: surface plasmon resonance, SPR. Measures real-time binding kinetics between the antibody and antigen. ELISA. Quantifies the ability of the antibody to bind to the antigen in a plate-based assay format. Flow cytometry. Assesses binding to cells expressing the target antigen, useful for cell surface targets. Functional characterization. Objective: to test the biological activity of the antibodies in relevant models. Process neutralization. Assays: determine the antibody's ability to inhibit biological activity of the target, for example, blocking receptor-ligand interaction. Cell-based assays: assess effects on cell signaling, proliferation, apoptosis, or other relevant cellular responses. Cross-reactivity and immunogenicity. Prediction. Objective: to identify potential off-target effects and predict immunogenicity. Process. In silico analysis. Use bioinformatics tools to predict cross-reactivity with human proteins and potential immunogenic regions. Western blot and immunohistochemistry. Test for binding to a panel of human tissues to identify off-target binding. Affinity maturation, ETE, if required. Objective: to increase the binding affinity of the antibody for its antigen. Process: directed evolution. Mutagenesis of the antibody variable regions, followed by screening for higher affinity variants. Rational design. Using structural information to introduce specific mutations expected to enhance binding affinity. Humanization for non-human antibodies. Objective: to reduce immunogenicity of non-human antibodies for therapeutic applications. Process: CDR. Grafting. Transferring the complementarity determining regions, CDRs, from a non-human antibody to a human antibody scaffold. Vernier zone engineering. Modifying framework regions to accommodate the CDRs without distorting their conformation. Stability and solubility testing. Objective: to ensure the antibody remains stable and soluble under physiological conditions. Process: thermal stability. Assays: determine the melting temperature, Tm, of the antibody to assess thermal stability. Aggregation tests. Use techniques like dynamic light scattering, DLS, to assess the tendency of the antibody to aggregate. Scale-up and GMP production preparation. Objective: to produce the antibody in larger quantities for further testing or clinical trials, adhering to good manufacturing practice, GMP standards. Process: clone selection. Choose high-expressing clones for scale-up. Process development. Optimize expression and purification processes for higher yield and purity. Preclinical testing. Objective: to evaluate the safety and efficacy of the antibody in vivo before clinical trials. Process: in vivo efficacy studies. Test the antibody in animal models of disease to assess therapeutic potential. Toxicology studies. Evaluate the safety profile of the antibody in animal models. Conclusion. The choice of technology for generating diversified oligonucleotide libraries depends on the specific requirements of the project, including the desired level of diversity, the complexity of the sequences, and the downstream applications. Each method offers unique advantages and limitations, and often a combination of approaches may be employed to achieve the best results in antibody, peptide, or nanobody library generation. Stay curious, stay innovative. I'm Luke McLaughlin. If you would like to learn more about this topic, consider reading our full-length articles on biotechnologyreviews.com, or consider our podcast series on YouTube or Spotify at our Biotechnology Reviews channel. Our articles are also available on our LinkedIn newsletter, Biotechnology Reviews.